Welcome students. Today let's have a discussion about a question paper. Actually, I have taken a university question paper for PharmD students. In which we can have a thorough discussion about the questions which they asked, which I have taken is from KUHS University question paper, December 2023. This question were made into two parts because it is in 3 hours question paper. So we are going to discuss all the questions and as well as the answers. So in which it needs some time. So students can listen to it and you can write down a clear assignment and submit to my office. Please be sincere to do this, as it will be helpful for you more. So here the question code 203326, and as per the question paper I have taken, if you see here, it is for second PharmD, regular and supplementary in December 2023, in which they have asked especially for Pharmacognosy and Phytopharmaceuticals. 3 hours question paper, 70 marks maximum. Answer all the question to the point neatly and legibly. So the student should read clearly, neatly you should write. Do not leave any blank pages between answers. And indicate the question number clearly and correctly for the answers in the margin space. Always follow this. It's very easy. And answer all parts of a single question together. Don't write 2-3 times putting the number in different pages, half question. That's what the instruction says. Leave sufficient space between answers. One answer finished means at least 2-3 lines you can give a gap and start the next question in the margin. Draw tables, diagrams, flow charts wherever necessary. And diagrams, especially pharmacognosy and phytopharmaceuticals, please make your diagram with pencil. Use pencil, scale and rubber whenever you are coming to an examination. And it is necessary for you to write the chemical structures, can be written by your pen and even pencil as you wish. Essay question, they used to ask 3 questions for PharmD students, 10 mark question. Number 1, they have asked, discuss the various method of adulteration of crude drugs with examples, 10 mark question. Discuss in detail about various types of classification of crude drugs, give examples. And number 3, explain the macroscopical and microscopical characters of clove bud and with labeled diagrams, with pencil labeled diagram, clearly macroscopy and microscopy. We will discuss this essay in this one lecture. 2 lectures are needed for the question paper to be discussed because 3 hours question paper. So I thought first let me discuss about these 3 essay questions, and next one more lecture with short notes, 8 questions with 5 mark, 40 marks they are asked in this question, December 2023. Describe the source, method and preparation and uses of absorbent cotton. Define proteins and classify with examples. Explain in brief on various factors affecting cultivation of crude drugs, give the chemical test to differentiate tragacanth and acacia, give the source chemical constituents and uses of chalmogra oil and olive oil, detail about the scope and development of pharmacognosy. 10th question, give the merits and demerits of natural pesticides. And the last question, 5-mark question, 8 questions they have asked for 5 mark, short notes. Enumerate the various cell constituents and cell inclusions. So we will discuss the short notes in the second lecture. First we will go for the discussion about the essay question, 3 10-mark question. First they have asked number 1, discuss the various methods of adulteration of crude drugs with example. We have class separately, we have done the class, but still I am explaining what to be written for the 10-mark question. Adulteration of crude drugs, start explaining about adulteration is an illegal act or debasing or alternating the original quality and properties of an article or a product in order to cheat or defraud others. In a broad sense, any change in an article that lowers its standard, quality or composition, irrespective of cause, it regarded as adulteration. The student should write very clearly about like this. And drug is regarded as adulterated if it does not conform to the standard or the quality of the genuine drug by whose name is sold or supplied. That is also adulteration. Crude drugs are adulterated by number of illegal acts. These various acts of adulteration may be roughly divided into 2 groups. One is undeliberately adulteration, without our knowledge, undeliberately. When the act of adulteration takes place due to various unintentional reasons, unintentionally adulteration, undeliberate adulteration. Next one is deliberate adulteration. When adulteration is done intentionally to cheat others, that is deliberate adulteration. So, 2 categories mainly you can differentiate. Undeliberate adulteration, the principal cause of undeliberate adulteration may be faulty collection. Faulty collection of plant drugs occurs due to wrong season, wrong age, less important parts collection, wrong identified and imperfect preparation. This all comes in undeliberate adulteration by in faulty collection. The collection of drug at a wrong season or at a wrong age of the plant, collection of less important parts of the plant due to ignorance, collection of drug parts or drug samples from both genuine and wrongly identified similar parts due to ignorance or inadvertence or inadvertence of the collector, or you can say a less knowledge of a mistake done by the collector. Imperfect preparation of the drug occurs due to the following factors: improper drying because use of wrong method of wrong temperature, incomplete garbling, removal of debris, imperfect garbling due to negligence and carelessness, improper packing. This all comes in the imperfect preparations. So this all comes under the undeliberate adulteration, incorrect storage also. In improperly built storehouses without proper ventilation, control of humidity, improper control of humidity, light and temperature. These various causes results in the production and supply of number of undeliberately adulterated crude drugs, which include inferior, deteriorated, spoiled and admixed drugs. An inferior drug is a substandard drug which contains less quality and active principles. An example you can say datura stramonium and datura metal mixing up. If you see, if the leaves of datura stramonium samples contain less than 0.25% of total alkaloids, they constitute an inferior stramonium drug, which is regarded as an adulterated one. So datura stramonium sample, when it is mixed with datura metal, which is having less constituents, is said to be an adulteration. A deteriorated drug is one in which the active constituents have been partially destroyed by overaging, moisture, heat, fungi and insects or by other means. Example of such drugs which are regarded as adulterated include hardened powder. Squill is an example, is a rodenticide. Squill, it looks like an garlic but it is not garlic. Hardened powder, due to aging, overaging, it becomes hardened and useless for the usage as a rodenticide. Over roasted coffee beans, which you are using coffee normally, roasted, more roasted, dark, charred and roasted. If you are more roasted coffee beans, they are also becoming and less quality and cannot be used. Moldly ergot. Ergot, you can say it is moldy. Other microorganism growth, which we can see in the picture. Original ergot, right? And moldy ergot. Wormy rhubarb. Rhubarb, undergrown stem, where you can see worms as shown in the picture. Wormy rhubarb, deteriorated drugs when you are supplying, that is also called as adulteration. Rancid lard. Lard, you can see the lard, the fatty substance, the becomes rancid. A crude drug is regarded as spoiled when its quality or unusefulness is so impaired or destroyed by the action of fungi, bacteria, insects and rodents as to render it unfit for the human consumption. All such drugs, when offered for sales or consumption, are legally considered as adulterated. An admixed drug is one in other parts of the same part of the plant or the same part of another similar plant, or get mixed through accidentally, ignorance or carelessness, they are also adulterated. Like senna, when you are mixing with senna pods as shown in the picture. Senna pods can be mixed with the dried leaves. or the stem part. Senna leaves containing excess amount of senna stems or pods would be an admixed drug, and thus it would be regarded as an adulterated. Different means of adulterating drugs. One is replacement of exhausted drug. Number 2, similar inferior drug. Number 3, artificially manufactured substituent drug. Substandard commercial varieties. And number 5, organic matter obtained from the same plant. And number 6, synthetic chemicals. So you should see the different means of adulteration of drugs can be done by means of writing very clearly. Replacement of exhausted drug, similar inferior drug, artificially manufactured substances, substandard commercial varieties, organic matter obtained from the same plant, and finally even synthetic chemicals. So when you come an example of first one, replacement of exhausted drug, which is very clear, particularly this is observed in the case of cloves, and saffron and tea and fennel, removing the volatile oil, can be sold, and ginger even. And these drugs are exhausted for their active constituents and reused with genuine drug after proper treatment. Exhausted. Saffron is a color artificially, while ginger is mixed with starch. Saffron sometimes it may be colored artificially and they can be sold at adulteration. Ginger is mixed with starch and colored to produce a proper shade. The fictitious nutmegs are made from mineral matter pressed into mold and flavored. So this is nutmeg, how they make it into pressed with some mold and made a flavor, and it is also can be replaced by the original drug. Number 2, similar inferior drug substitution with similar but inferior drug. The harvesting of cultivated drug when has been treated minimum standard of quality yields inferior drug. The common example of substitution is adulteration of clove by mother cloves, which is having less constituents. Saffron is adulterated with dried flowers of Catranthus tinctorius. Safflowers like this can be adulterated. Digitalis containing very less amount of glycoside is used to dilute with the preparation and prepare digitalis leaves containing higher percentage of glycosides, like digitalis lanata with digitalis purpurea. They both are very good quality. Other than that, there are many low variety species which can be mixed up. Number 3, artificially manufactured substituents. Substituents of artificially manufactured substituents, this type of adulteration is observed in case of drugs which are costly. Paraffin wax is tinted yellow and substituted for yellow beeswax, which is very costly. So it can be mixed up artificially, while artificial invert sugar is mixed with honey. It is also done as an adulteration as shown in the picture. Number 4, substandard commercial varieties. Substitution by substandard commercial varieties. Red chilies, example of capsicum frutescens with capsicum minimum are substituted by capsicum annum, while gentian luteae is substituted by Picrosia corera. You see this capsicum, how they are adulterated, like Gentium lutea can also be substituted by Picracia cororia or corora. The substitution of Alexander senna with Arabian senna as shown in the picture, and substitution of rhubarb with many other species of rumex like as shown in the picture is possible. And other examples like Naxomica seed, Naxomica, Strychnus naxomica, adulterated with Strychnus naxbunda or Strychnus potatorum seed. Strychnus naxomica is this one. Strychnus potatorum is these seeds. They can be adulterated. And Strychnus naxbunda also can be substituted. Number 5, organic matter obtained from the same plant. The presence of organic matter obtained from the same plant in case of advantages of similar color, odor and constituents, it takes into consideration and other parts of the same plant are added to the genuine drug. Clove is mixed with clove stalks. See that clove stalks, color is same, flavor is same, but the constituent of eugenol may not be present there much. So they can be substituted, they can be added with that. While caraway and anethum fruits, the Umbelliferaceae fruits are mixed with other inflorescence. It can be mixed caraway, anethum fruits, dill. They are added with their other inflorescence. Inflorescence means other parts. Like a pedicel can also be added with this one. 6th one is synthetic chemicals. Many types of synthetic chemicals which constitute one of the chemical constituent of the drug is added to the genuine drug. Example, benzyl benzoate to balsam of Peru and citral to oil of lemongrass and camphor oil and eucalyptus oil in oil of rosemary. This all can be adulterated by means of benzyl benzoate. And you can see several times waste from the market are collected and admixed with drugs. This is observed in case of unorganized or liquid drugs. The piece of amber colored glasses in colophony, you can see the picture, limestone in asafoetida, Kayam, where they used to add limestones, white oil or oil of coconut and stearine or paraffin, cocoa butter are deliberately added kind of adulteration. They will be added. These are deliberately adulteration, adding limestone in asafoetida, glass, amber colored glasses pieces in colophony, paraffin in cocoa butter, styrene, this all should be added. The question number 2 is 10-mark question. Discuss in detail about the various types of classification of crude drugs. Give examples. Listen to it. Make clear for a 10-mark what should be written. Clearly write all the points. You will get the full mark. Here, classification of crude drugs. Students should write, classification can be done by alphabetic classification, morphological classification, taxonomical classification, pharmacological classification, chemical classification, chemotaxonomical classification, serotaxonomy. These all 7 you should explain with examples. That is the question of 10 mark. Alphabetic classification means, alphabetically classification is the simplest way of classification by any disconnected items, crude drugs are arranged in alphabetical order, or that's Latin or English name, common name, or sometimes local names, vernacular names. Some of the pharmacopoeias, dictionaries and reference books which classify crude drugs according to this system as follows: Indian Pharmacopoeia, British Pharmacopoeia, British Herbal Pharmacopoeia, United States Pharmacopoeia and National Formulary, British Pharmaceutical Codex, European Pharmacopoeia. In European Pharmacopoeia, these are arranged according to the names in Latin, where in USP and British Pharmacopoeia BPC for Pharmacopoeia Codex, these arranged in English. Merits: it's easy and quick to use. There is no repetition of entries in void of confusion. In this system, location, tracing and addition of drug entries is easy. Demerits: there is no relationship between the previous and successive drug entries. Examples like you can write acacia, benzoin, cinnamon like C, D, and dill, E for ergot. That's an example. I am just writing in alphabetical order. You can write gentian, hyoscyamus, ipecacuanha, jalap, kurchi, licorice, mints, naxomica, opium, podophyllum, quassia, rauwolfia, senna, vasaka, wool, that means wool fat, yellow beeswax, zeardry. Like that you can have different. Zeardry means actually white, actually turmeric, skin inflammation scars which we are using, especially the curcumin. So like that you can have the examples. It's also called as white turmeric, perennial herb from the ginger family, Gingiberaceae, zeordry. Just gave the explanation. Morphological classification. This is a system of drugs which is arranged according to the morphological or the external characters of the parts of the animal parts, plant parts or animal parts. Which the part of the plant used, like example leaves, roots, stems, etc. The drug obtained from the direct plant parts of the plants and containing cellular tissues are called as organic drugs, like rhizomes, barks, leaves, fruits, entire parts, hairs and fibers. The drugs which are prepared from the plants by some intermediate physical process such as inclusion, drying or extraction with a solvent not containing any cellular parts or cellular plant tissues are called as unorganized drug. Aloe juice, opium latex, agar, gambir, gelatin, tragaganth, benzoin, honey, beeswax, lemongrass oil, examples of unorganized drugs. So organized drugs, example wood, for example quassia, sandalwood, red sandalwood, this is quassia. Leaves, digitalis, eucalyptus, gymnema, mint, senna, spermamint, squill, tulsi, vasaka, coca, buchu, hamamelis, hyoscyamus, belladonna tea. Bark, Arjuna, Ashoka, cascara, cassia, senna, cinnamon, kurchi, quillia, wild cherry. Flower parts, clove, pyrethrum, saffron, santonica, chamomile. Fruits, amla, anise, bael, behara, bitter orange peel, capsicum, caraway, cardamom, colocynth, coriander, cumin, dill, fennel, gokuru, hedra, lemon peel, cinnamon, star anise, tamarind, vidam. So these all seeds examples: bitter almond, black mustard, cardamom, colchicum, ispagulla, caladana, linseed, nutmeg, naxomica, phytosigma, Phyllium, Strophanthus, white mustard. These are all the examples for organized drug. Roots and rhizomes, example, aconite, ashwagandha, calamus, kalumba, colchicum, corn, dioscorea, galanga, garlic, gentian, ginger, ginseng, glycyrrhiza, podophyllum, ipecac, ipomia, jalap, jatamansi, rauwolfia, rhubarb, sasuria, seniga, satavari, turmeric, valerian, squill. Plant and herbs, the whole herbs: ergot, ephedra, bacopa, Anthrographis paniculata, Kalmeg, yeast, Vinca, Datura, Centrella. So these are some of the examples: hairs and fibers, cotton, hemp, jute, silk, flax. So all of them are organized drug, parts of the plant or the whole plant. Unorganized drug, they are derived from the plant: dried latex, opium, papain, dried juice, aloe, quinoa, dried extract, ago, alginate, black catechu, pale catechu, pectin, waxes, beeswax, spermaceti, carbowax, gums, acacia, guar gum, Indian gum, sterculia, tragaga. Resins, asafoetida, benzoin, colophony, copaiba, guacum, gucul, mastic, coal tar, tolu balsam, storex, santorac. Volatile oils, examples like terpenes, anise, coriander, peppermint, rosemary, sandalwood, cinnamon, lemon, caraway, dill, clove, eucalyptus, nutmeg, camphor. Fixed oil and fats: arachis, castor oil, chalmogra oil, coconut oil, cottonseed oil, linseed oil, olive oil, sesame oil, almond oil, theobroma oil, cod liver oil, halibut liver oil, cocoa butter. These all fixed oil and fats example. Animal product: beeswax, catherides, cod liver oil, gelatin, halibut liver oil, honey, shark liver oil, shellac, spermaceti wax, wool fat, musk, lactose. Fossil organism and minerals: bentonite, kaolin, Kesselgar, talc. Difference between organized and unorganized drug. When you see organized drug, these are plant or animal origin. Unorganized drug, they are plant, animal, or even it can be a mineral origin. Organized drug, these are direct part of the plant or animal. Unorganized drug, they are products of plants or animals. Organized drug, these are cellular structure. Unorganized drug, these do not have well-defined cellular structure. Organized drug generally identified by morphological characters. Unorganized drug generally identified by organoleptic properties. Examples of organized drugs, you can say digitalis leaf, cinchona bark, and ephedra stem, whatever maybe we have previous slides we have seen many. Examples for unorganized drug, agar, gelatin, honey, which we can give the example. Merits of these morphological characters are more helpful to identify and to detect adulteration. This system of classification is more convenient for practical study, especially when the chemical nature of the drug is not clearly understood. Demerits. The main drawback of morphological classification is that there is no co-relationship to the chemical constituents with the therapeutic actions of the particular drug. Taxonomical classification is an interesting old classification. Is a purely botanical classification based on the principle and natural relationship of the evolutionary development. They are grouped by kingdom, phylum, order, family, genus and species. As all the entire plants are not used as a drug, part of the plant is used as a drug, for example cinnamon bark. Thus it is no significance in identifying point of view, put the plant in taxonomical order. But anyhow, we should know about Talophyta means Talophyta or Talobionata are a polyphyletic group or non-moiety organized, traditionally described as thalloid plants. Algae is an example. You can see algae, Talophyta. Pteridophyta. A pteridophyte is a vascular plant with xylem and phloem that reproduces or reproduced by means of spores. Because Pteridophyta produces neither flower nor seeds. Example, fern, male fern, which we might have seen in an ornamental plants which we are using. Gymnosperm and vascular plant, any vascular plant that reproduces by means of exposed seeds or ovule, unlike angiosperms or flower plants whose seeds are enclosed by the mature ovaries or fruits. Example, ephedra, which we are doing in the lab. You should know that examples of gymnosperms. Angiosperm are the plant that produce flowers and bear their seeds in fruits. Example, opium, fruits bearing the seeds. So, we have discussed about this Talophyta, Pteridophyta, Gynoecium, Gymnosperm and Angiosperm. These 4 points you have to remember with example. The main characters of various taxon that contribute crude drug plant kingdom. Pteridophyta, algae and fungi are considered together. They differ in mode of nutrition, algae exhibit autotrophic and fungi exhibit heterotrophic nutrition. Example, Rhodophyta, red algae, agar. Pteridophyta, this group of plant derives in the name of fern, Petris, which also represents the salient features of group Pteridopretis or phytone plants. They occur in humid and tropical climates and usually ground or soil, rocks in ponds, etc. These plants also rise in the ponds of the ornamental plants, example male fern, like Pteridophyta. Gymnosperm, the gymnosperm we know that is gynous, naked, and sperma, seeds of plants with naked seeds. The living gymnosperms are widely distributed in the cold climates. These are seed producing plants. Seeds are not enclosed in the fruits. Example, ephedra. Colophony includes conifers, pine, scrubs, ferns, cycads, ginkgo, and genitophytes. Angiosperm, the term angiosperm means includes seeds because of ovules and potential seed are enclosed with hollow ovary. The angiosperm consists of most dominant ubiquitous vascular plant present day flora. Dicotyledon and monoclots are the subdivisions of this angiosperm, which we have seen that examples. Seed wings, this is about the conifers. Just to show you the seed wings of conifers, seeds are not inside the fruit. Wings of the seeds, which we can see the conifers, pine seeds, just to show you the picture. This is about the ephedra. And the monocot, they are having two cotyledons, example vanilla and colchicum. Monocot, this is the picture. Monocot refers to the group within angiosperms or the flowering plants characterized by having a single cotyledon or a seed leaf in their seeds. So, parallel veins in the seeds can be seen. Scattered vascular stem and multiples of 3 in the flowers, and fibrous roots can be seen in monocot. Dicot, they are two cotyledons, example coriander, capsicum, like two cotyledon seeds. And in this type, the flowering plants characterized by two seed leaves, net like leaf venation, and floral parts typically in multiple of 4s or 5s as shown in the picture. Taproots, branches of veins, leaves will be as shown 5 to, and petals 4 to 5 petals, flowers, and the stem will be ring shape as shown. The vascular bundle, how the rings of xylem and phloem, cambium can be seen in this very clearly in the dicot plants. So this is monocot, and the roots of fibrous roots, parallel veins, multiples of flowers, petals, and having scattered vascular bundles. Dicotyledons is two cotyledons with taproots, branch veins in the leaves, and 4 to 5 petals of flowers, and a ringed vascular bundle as shown in the stem area of cambium, which we know about the stem. Taxonomical classification of some crude drugs. Phylum, examples I have taken: Talophyta, bacteria, fungi, lichens, Rhodophyta. Examples of Gelidiaceae, agar is a drug. Pteridophyta, liverworts, moss or ferns. Felicalis, Polypodaceae, male fern is an example of the drug. Gymnosperms, genitals, Ephedraceae, ephedra, coniferae, Pinnaceae, colophony. And example for angiosperm, monocotyledons, Lilliflorae, Liliaceae, colchicum. Microspamiae, Dioscoreaceae, Dioscorea. Orchidaceae, vanilla, example of drug. This is the picture of red algae, male fern, ephedra, colophony, and about how it is collected as an exudate or as an unorganized drug. And this is about the colchicum and Dioscorea, and this is about the picture of Dioscorea leaves, and this is about vanilla. Now angiosperm dicotyledons example, Pepperless, Pepperoraceae, opium is a drug. Rosalis, Rosaceae, almond. Malvaceae, sida. Order, example I am telling the reading first. Tubiferae, Convulsulaceae, Labiatae, Solanaceae, Scrophoreraceae, like Shankupushpi, Mentha, Belladonna, digitalis for each family. Plantagenes, that is Plantageneaceae, example Plantago, Plantago ovata, that is the drug actually. Ispagul or linseed, they all comes under there. And Campanulus or Lobelia, family Lobelia. This is the example of opium plant, and this is about almond, and this is about sida, and this is, we know that coriander, and we know about the scaraway, and the fennel which comes in umbelliferaceae fruit, Shankupushpi, and this is about mentha, belladonna, digitalis, and this is Plumpago ovata, and this is about Lobelia. Merits taxonomical classification is very helpful for studying evaluation and development. Demerits, this system also does not have co-relationship between the chemical constituents and the biological activity of the drugs. The next classification is pharmacological classification, grouping of drug according to their pharmacological action or of the most important constituents of their therapeutic uses in terms of pharmacological or therapeutical classification of the drug. This classification is more relevant and it is most followed method. The drugs like digitalis, squill, strophanthus having cardiotonic action are grouped together irrespective of their parts used or phylogenetic relationship or the nature of the phytoconstituents they contain. So classification is on the basis of the pharmacological action. Just an example I am giving, like anti-cancer, vinca, podophyllum, taxus, anti-inflammatory, colchicum, turmeric, anti-amoebic, ipecacuanha or ipecac root, kurchi bark, anti-asthmatic, ephedra, lobelia, anti-helminthic, male fern, quassia wood, antispasmodic, datura, hyoscyamus, astringent, example catechu, and analgesic, example opium, poppy, bitter tonic, example quassia wood, naxomica, gentian, carbonative, example coriander, fennel, clove, peppermint, purgatives, example senna, rhubarb, Expectorant example tulsi, balsam of Tolu, Vasaka. Cardiotonic example digitalis, squill, strophanthus. Tranquilizers example Rauwolfia roots. Like that you should write an examples for each and every classifications for 10 mark questions. Merits. This system of classification can be used for suggested or suggesting substituents of the drugs in these are not available at a particular place or point of time. If they are not available, it's possible by means of their activity. Demerits. Drugs having different action on the body gets classified separately is more than one group can cause ubiquity and confusion. The same activity may be seen. Cinchona is anti-malarial drug because its presence of quinine. But at the same time, the same drug contains quinidine, which undergo affecting the heart because it is anti-arrhythmic action, quinidine present. So cinchona have to be written in two places. So that's the confusion. Chemical classification. The crude drugs are divided into different groups according to the chemical nature of their most important constituents. Since the pharmacological activity and therapeutic significance of a crude drug are based on the nature of their chemical constituent, the chemical classification of the drug dependent on the group of drugs with their identical constituents. And out classification as follows: carbohydrates, polyhydroxy, aldehydes, ketones containing an unbroken chain of carbohydrate atom. Examples of gums: acacia, tragaganth, guar, mucilage, plumbago seeds, and other seeds like other starch containing drugs like honey, agar, pectin, cotton. Like that you can classify. Glycosides are containing group compounds, hydrolysis, which rise to sugar, aglycan and glycan molecules like anthraquin glycosides, aloe, cascara, rhubarb, senna, saponin glycoside, quillia, arjuna, and glycyrrhiza glabra. Cyanophore glycosides, wild cherry bark example, isocyanate glycosides, mustard, and cardiac glycoside digitalis, strophanthus example, bitter glycosides, gentian, columba, quassia, charata, calmec examples. And number 3, you can see tannins are the complex organic non-nitrogenous derivatives of polyhydroxy benzoic acid example, pale catechu, black catechu, ashoka bark, galls, myrobalan, bahera, amla examples. Volatile oils, monoterpenes, sesquiterpenes obtained from plants like cinnamon, fennel, dill, caraway, coriander, cardamom, orange peel, mint, clove, valerian. Lipids like fixed oils, castor oil, olive oil, almond oil, shark liver oil. Fats like theobroma, lanolin. Waxes like beeswax, spermaceti, and resins. Complex mixture compounds like resinols, resin acids, resinotanols, resinins. Examples: colophony, podophyllum, cannabis, jalap, capsicum, turmeric, balsam of Tolu, Peru, asafoetida, myrrh, ginger. And number 7, alkaloids, nitrogenous containing plant origin compounds, pyridine and piperidine derivatives of alkaloids, lobelia, nicotiana, tropane alkaloids, example coca, belladonna, datura, stramonium, hyoscyamus, henbane, quinoline alkaloids like cinchona, isoquinoline alkaloids like opium, ipecac, columba, indole alkaloids, ergot, Rauwolfia, amine alkaloids like ephedra, and the purines like tea, coffee, and number 8, proteins, examples like gelatin, fisin, papin, and vitamins from yeast, and terpenes like rasna, colocynth. And the merits of this type of classification, chemical, it is a popular approach of phytochemical study. Demerits, ubiquitous rises in particular drug process, number of compounds belonging to different groups of compounds may be present in the same plant is also possible. Chemotaxonomical classification, this system of classification relies on chemical similarity of a taxon. It's based on the existence of a relationship between constituents of various plants. There are certain types of chemical constituents that are characterized certain classes of plants. This gives birth to entire new concept of chemotaxonomy and utilizes chemical fact, characters of understanding of taxonomical status relationship with an evolution of the plants. For example, tropane alkaloids generally occurs among the members of Solanaceae family, thereby serving as chemotaxonomic marker. Similarly, other secondary plants metabolites can serve as basis of classification of crude drugs. The berberine alkaloids, berberis, and argimonon, routine of Rutaceae members, Ranunculaceae alkaloids among the members, etc. are other examples. It is latest system of classification and gives more scope for understanding the relationship between chemical constituents, their biosynthesis, and their possible action. What is serotaxonomy then? Define serotaxonomy. They used to ask in short notes. Serology is defined as that portion of biology which is considered with the nature and interaction of antigenic material and antibodies. Smith 1976 defined it's a study of origin, properties of antisera. Serotaxonomical classification. The serotaxonomy can be explained as a study about the application or an utility of serology is solving the taxonomical problem. Serology can be defined as the study of antigen-antibody reaction. Antigens are those substances you can say, which can stimulate the formation of antibodies, antigens. The antibodies are highly specific proteins, molecule produced in plasma cells in immune system. Proteins are carrier of the taxonomical information and commonly used as an antigen in serotaxonomy. For serotaxonomy works, protein extraction. Proteins are extracted from the plant tissue like seeds. Antigen-antibody reaction studied. The extraction protein are injected into the animal to induce the production of antibodies against them. Blood samples are collected. The samples are collected from the animal which contain the antibodies against the plant protein. Comparison. The blood sample is used to compare with the first plant protein with proteins extracted from other plants. If the antibody reacts strongly with the protein from the second plant, it suggests that it is closer evolution relationship between the two plants. Example of serotaxonomy in plant taxonomy: Magnolidae family, Hamimelidae family, and Comiflorae family. They all three serotaxonomical study that seed protein indicates close relationship among these groups, suggesting a common ancestor rather than independent evolution. Actually, this is a picture to show you how the extract is taken, injected into the animal, an antigen and antibodies are produced. These monoclonal antibodies will be used in the petri dish plates, and we can incubate it and we can find the reaction with the other plants, a sample which we have taken. And so that we can find out the relationship. If they have the antibody antigen reaction shows, they have the relationship of the extract. So this is a new technique of classification by means of serology. That's why it is called serotaxonomy. Number third, the 10-mark question. Explain the macroscopical and microscopical character of clove buds with labeled diagrams. All diagrams should be drawn with pencil in your examination point of view. Clove, we know that, is an important plant which is more grown in Kerala and in our area. Cloves are dried flower buds, Schizogenium aromaticum, Eugenia caryophyllus, Schizogenium aromaticum, and its another name of a botanical name is Eugenia caryophyllus, family Myrtaceae. Geographical source, it is native from the Molucca island and traditionally cultivated in Tanzania, Madagascar, Indonesia, Sri Lanka, India mainly Nilgiris Hills, and in Kerala, Kanyakumari, Kottayam, Koilon Hills of Kerala. Here, cultivation and collection. The deep, rich, loamy soil is good for clove cultivation, but it can be grown in sandy, loamy and laterite soil. It favors warm, humid climate and annual rainfall in a range of 150 to 250 centimeter. It grows well in an altitude of 90 or 900 meters from the sea level. The seeds are sown in August or October for cultivation purpose. Seeds are sown in nursery bed in distance of 10 centimeter. The seeds germinate in 4 to 5 weeks, and these seeds are transplanted to a pot for 6 months. These seedlings are kept in a pot for nearly about 12 months. Then they are transferred into open field. The field is provided in initial stage because the plant cannot bear the full sunshine. It should need some shade, and it has to be cultivated along with other plants like arecanut, coconut or nutmeg. The suitable fertilizers like ammonium sulfate, superphosphate, potash, etc. can be provided in the plants to 2 doses. The first dose should be given in the May or in the June, and the second dose will be given in the October. The plant started bearing 7 to 8 years. After 8 years, they give abundant yield of hectares after 15 to 20 years of growth. Generally, the clove plants produces 3 to 4 kg of drug per year. The clove buds are hand-picked and beaten with bamboo and sometimes platform, ladders, and also used by case in tall trees. The collection starts from the clove buds, changes into color from the green to light pink as shown in the picture, and the cloves are sun-dried and kept free from the foreign matters. Like as shown, the color will be changed. After drying, the color of the clove changes, become crimson or brown black as shown in the picture. And we can see that crimson red, color is crimson red to brown. Macroscopical characters as the question asks. Color characteristic or aromatic odor, taste aromatic and pungent. Size is length of 10 to 18 millimeter, width is 3 to 4 millimeter, and thickness is 2 millimeter. Shape flower, buds are nail shaped, hypanthium quadrangular, 10 to 12 millimeter into diameter 2 to 3 millimeter. Head, globrous. Hypanthium, the hypanthium, neck part, corresponds to the inferior ovary and globulous head, and surrounded by 4 divergent petals. Sepal lobe consists of 4 imbricated petal wrapped numerous inchord stamens. The pulverous drug shows a presence of large secretory cavities, calcium oxalate crystals, pollen grains, fibers with thick lignified puncculate walls. This is the diagram you have to draw very clearly to show the epidermis, oil glands, vascular bundle, colonchema, parenchyma cells and cortex, and should be shown very clearly the 45x diagram with cuticle, epidermis and rhaphides, the calcium oxalate crystal cells and vascular bundle, pericyclic fibers and parenchyma cells and colonchema, the middle cells, to be shown TS very clearly for microscopy. Clearly you should explain the layers. I have given only the names. You should explain, as it is a 10-mark question. Heavy cuticle epidermis, numerous oil cells, lysogenous, you should specify that. Calcium oxalate crystal clusters, crystals of prisms, stomata, epidermis of petals, starch, fruit of mother clouds, and lignified sclerenchyma cells. You should explain this picture as well as the names and the parts of the picture. Constituents: 14 to 21% of volatile oil, mainly eugenol, isoeugenol, cetosterol, stigmasterol, camphosterol, tannins, terpene, acids and esters are present, glycosides. The main constituent is eugenol. You should write the structure: benzene is OH, OCH3, CH2, CH, double bond CH2. Clearly you should mention this eugenol structure. Only one structure is, if you write, it will be good for, and it will be supportive for giving the full mark. Properties of clove oil, very interesting. Color: the clove oil is colorless to pale yellow. Specific gravity is 1.038 to 1.06. Refractive index 1.527 to 1.535, and boiling point 250 degrees centigrade. It should be stored in a proper container or conditions because dark and thick on storage. And chemical test: needle shaped crystals are seen when a section of a clove is treated with a strong potassium hydroxide solution. Uses: you know that clove, based on the phytomedicine, are used locally to treat minor wounds and cleansing, as an analgesic, headache, toothache, and an analgesic in disease of mouth, that is pharyngitis, pharynx, and both in the form of lozenges and mouthwashes and oral hygiene. Internally, it is used in the treatment of symptoms of gastrointestinal disturbance like epigastric, bloating, improper digestion and flatulence. In Germany, clove-based products are used in mouthwashes for swelling of mouth and throat. In Indonesia, it is consumed as a cricket cigarette. An adulteration of the clove. Exhausted clove, usually they are adulterated deliberately. So oil-free, containing less amount of oil, dark in color, shrink and floats on the water. Sometimes clove stalks are adulterated. Generally used adulterated powdered clove or ash value will show high, and fiber content of high. Calcium oxalate prisms are present containing 5% of oil. We can evaluate and you can find out. Blown cloves also, expanded flowers of clove trees, stamen free, containing less volatile oil. Mother clove, dark brown in color, oval shape, bigger ones, light, aromatic, and starchy containing less volatile oil. These are some of the adulteration which can be done. So we have discussed about the essay question, 3 essay question, and we have to continue for the short notes. There are 8 short notes are there which we will discuss now. I think it is the class is we need a gap so that after the next hour we will take the continuation. Thank you.